What is SNP? SNP means single nucleotide polymorphism. Single nucleotide polymorphism means a single nucleotide difference at the DNA level, at the gene level. It can be differentiated between individual to individual or between different individual of a given population. So this is a molecular technique that is we can observe this marker. This is the marker means just to point out where there is a difference in the genome. Suppose if you take the human body, in human body approximately 4 to 5 billions SNP, such type of difference are there in their genome. And all over the world of population in the human, there is a 100 millions SNP spot, means the difference of nucleotide in the individual that can be detected. So this SNP generally we can find after 1000 nucleotide in human genome, or on an average we can also observe 200 to 300 nucleotide after 200 to 300 nucleotide. So this is the most important technique just to predict the variation of two different twins, two different clones. Means we can find out the variation, we can find out the variation of the uh, among the individual, because 99.9% genome of human is are identicals. Only 0.1%, up to 1% variation occurs at the gene level, due to which different individual looking different, different individual looking different. The face of that individual will be differ from that individual, and the face of that individual also differ with that individual. So this SNP, that is a technique through which we can differentiate of the individual of a given population. Means if you want to study the difference of the different individual, they, if there is a single nucleotide difference, even then we can detect with the help of SNP. So try to know what is why SNP is required. Why SNP is required? SNP that is generally used for such, find out the such type of variation. First point. Second point, if you want to observe the population, whether it is the identical clone population or there is a difference or variation in the gene level of a population, that things can be studied with the help of SNP. Third point, that is the, if a gene is associated with the defective, means the, if there is a defective gene present or regulatory gene that is associated with the defective gene, then there is a disease development. Such type of
सर
study, हाँ बोलिए।
सर ये सर दो बच्चे सर कॉल करके मेरे को कह रहे हैं कि सर को बोलो एडमिट कर दें हम दोनों को भी क्लास
अच्छा
के लिए।
ठीक है ठीक है। सो ये मैं कहना चाह रहा हूँ कि SNP, इट इज, व्हाट इज द फुल फॉर्म ऑफ SNP? इट इज द सिंगल न्यूक्लियोटाइड पॉलीमॉर्फिज्म। इट इज द सिंगल न्यूक्लियोटाइड पॉलीमॉर्फिज्म। So we can differentiate the single nucleotide level difference of at the gene level of the different individual. We can easily understand with the help of this molecular technique that is known as SNP. SNP, thus this diagram actually indicate the position, means where the difference is are occur at one through one nucleotide. So in the two or three person, they are differ from each other, all are identical. You see, you can observe all are identical. This is GCA, 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 all are similar. Like CGTTAGA. So these are similar in nucleotide sequence, but only one nucleotide difference are there in one position of the genome of one individual. So they are differ each other only due to one nucleotide. Such nucleotide difference that can be studied with the help of SNP. What are the application of SNP? SNP are used for find out the variation among the individual to individual of a given population. Means suppose there is a twins or there is a clones. In that situation also we can differentiate of one nucleotide or two nucleotide. If there is a millions, if there is a hundred individual population, so that can be differentiated if it is human. You know, human population, human genome is from one to other that are exactly 99.9% similar, but there is only 1.1% difference. Up it can be vary up to 0.2% difference, due to which individual are differ from each other from their at the genetic level. Means they are differ in appearance wise, they are differ in look wise due to difference of 0.1 to 0.2% genome. Okay, so this is the major application just to find out the variation of the SNP. So SNP, that is the due to one nucleotide difference. Second point, if a person have a defective gene, how can you find out the defective gene? With the help of the SNP techniques, or the some gene, that regulatory gene that control the defective gene. So the regulatory gene, the defect in the regulatory gene also can be identified with the help of SNP. Okay, second point. Third point, that is generally the SNP map. If you study the SNP map in your CBSE textbook, there is a given by bar diagram. Okay, so if you see the SNP map, if you see open the book of your CBSE textbook, so you just see, ये दिख रहा है आपको, ये जो है आपका SNP मैप है. सो वर्टिकल बार, दैट इज द वर्टिकल बार, ये दिख रहा है आपको, आपके बुक में है. ठीक है, ये दो. सो दिस इज द वर्टिकल बार. सीधा दिख रहा है क्या? ये दिख रहा है सीधा, ये दिख रहा है, ये आपके बुक में है. So generally this vertical bar indicate the position of SNP. SNP means the difference of one nucleotide. What is the frequency in the genome of human beings? It will occur after 1000 base pair. You can find the difference of one nucleotide between two individual or three individual after 1000 base pair. So just try to know the vertical bar in your book, CBSE textbook, that is the SNP map. So generally the SNP map give the clue or predict the effect of the response of the drugs of the doctor prescribe the medicine. The same medicine can affect differentially पेशेंट टू पेशेंट, कि फर्स्ट पेशेंट में ये जो है इसका हायर डोज चाहिए, सेकेंड पेशेंट में इसका लोअर डोज चाहिए, मीन सेम मेडिसिन आई एम टॉकिंग, और थर्ड पेशेंट में इसका जो है मीडियम डोज चाहिए. तो ये जो इफेक्ट है रिस्पॉन्स ऑफ द ड्रग, how can doctor predict what type of what dose of medicine can be given? It should be given by a doctor for treatment of particular disease with observing the SNP मैप. SNP मैप को ऑब्जर्व करके आप डॉक्टर जो है प्रिस्क्राइब करता है कि what will be the dose of the medicine. So response of the drug, that is the third point, that is the application of SNP. Means the commercial application of the SNP map is just to monitor the response of particular drug against different patients. Means they are these drugs at particular dose are effective in particular patient, and this same drug are effective at lower dose in the other patient. So thus such type of response can be monitored by the doctor with the help of SNP. Third point, fourth point, susceptibility of the particular disease. Suppose there are so many diseases that is associated with single nucleotide difference. You know, in your CBSE textbook, just if you go through the page number 63, so there is cystic fibrosis, there is cystic fibrosis disease, okay, in the left side there is cystic fibrosis disease. So cystic fibrosis disease that is associated with the CFTR gene, CFTR gene. So this CFTR gene, there is a defect in the CFTR gene due to which there is cystic fibrosis. The CFTR gene is present on the chromosome number 7. This CFTR gene is present on the chromosome number 7. So if you see there is a deletion. What is the deletion? Deletion of the codon of the tryptophan on the chromosome number 7. Means in case of CFTR disease, there is a defect of the gene. What type of defect? There is a deletion. There is a deletion of what? The gene responsible for tryptophan. So due to which there is a disease, that is the due to single gene mutation, that is cystic fibrosis disease. Next is Huntington disease. Huntington disease that is associated with chromosome number 4. It is given in your CBSE textbook. I am talking about your CBSE textbook, as per your CBSE textbook. So it is due to increase the number of copy of CAG repeat, in chromosome number 4. You can find 35 times repeat of CAG, that is the codon, CAG repeat. So due to which there is a Huntington disease. So these are the example of the CF, that is the SNP, the disease due to single mutation. So such type of single mutation can be detected. That is the cystic fibrosis type of disease and Huntington disease can be detected. Next is the Alzheimer disease. Next is Alzheimer disease. So Alzheimer disease is associated with chromosome number 19. It is associated with chromosome number 19 and chromosome number 10. So what is the gene is associated with the Alzheimer disease? Due to defect in APO gene, apolipoprotein gene. You must know the remember the gene name, APO gene, APOE gene. So it is given in your CBSE. I'm just going through your CBSE textbook. Just go through the table. Table number 2, page number 63. So you must go through the CBSE textbook only. So this is the gene, APO gene. There is a defect. So due to defect of APO gene, there is a disease that is the Alzheimer disease. So this is the world famous disease, that is the genetic disease, and this is due to defect in the chromosome number 19 and chromosome number 10. Next is migraine. You know migraine, there is a pain in the brain, okay, in the one side only. So this is also due to a defect in the gene, the chromosome number 1. So this is also a case of SNP. We can study this thing, these all disease with the help of SNP. कौन कौन सा डिजीज है आप बता सकते हो? पहला है सिस्टिक फाइब्रोसिस, ये क्रोमोसोम नंबर 7 पे एसोसिएटेड है. दूसरा हंटिंगटन डिजीज, ये क्रोमोसोम नंबर 4. आई एम टॉकिंग अबाउट ह्यूमन बींग्स. एंड नेक्स्ट इज that is Alzheimer disease. Alzheimer disease किससे रिलेटेड है? Due to defect of the APO gene, apolipoprotein gene. I'm talking about gene. It is a protein that is coded by this APOE gene, and due to defect of that we can find the disease, and this is associated with the chromosome number 19 and chromosome number 10. Next, that is the migraine. It is also associated. Next, you can also study the uh, uh, chemo chemokine receptor gene, CRR5 gene, chemokine receptor gene, that is due to that active receptor there is a disease like AIDS HIV virus. receptor. So this is chemoreceptor gene. If there is a mutation in that gene, then there is no chance of that disease. क्या हो सकता है? If there is a mutation, किसके में mutation हो जाएगा? CCR, chemokine receptor gene, CCR5, chemokine receptor gene, CCR5 gene, जो कि ये receptor है just to recept the HIV virus, just like coronavirus. Okay, so that is, if there is a mutation occur in that situation, the person become resistant to that virus. Okay, so these things can be studied with the help of SNP. That is the vast application of this SNP. So we can find the first point, just application. I'm talking about application of SNP, a study of different disease. कौन कौन सा डिजीज है? अल्जाइमर डिजीज है, दूसरा है हंटिंगसन डिजीज है, तीसरा है आपका सिस्टिक फाइब्रोसिस डिजीज है, चौथा क्या है कि दैट इज द माइग्रेन. वी कैन स्टडी विद द हेल्प ऑफ एसएन, means the gene associated with that will become defective. लेकिन CRR5 gene, that is the gene responsible for, that is chemokine receptor gene, that is, if there is a deletion occur, in that situation that person become resistant to HIV virus. मीन उसमें HIV virus, क्योंकि that is the receptor for HIV virus. Okay, so that the such thing can be studied with the help of the SNP technique. दूसरा, second point, we can find out the variation in the क्लोनल पॉपुलेशन. क्लोन है तो उसका वेरिएशन कर सकते हैं, कि अगर एक न्यूक्लियोटाइड का भी डिफरेंस है तो उसको हम स्टडी कर सकते हैं विद द हेल्प ऑफ this SNP maps. Okay, or SNP techniques. Okay, third, if you want to find out, means variation to differentiate the twins, कि एक न्यूक्लियोटाइड का डिफरेंशिएट है, फिनोटाइपिकली दे आर सिमिलर but genotypically they are different. Such difference can be studied with the help of this SNP. So these are the application part of the SNP. Now come to what are the different type of SNP. So generally ये SNP आपको पता चल गया होगा कि SNP क्या है, कि जस्ट वन न्यूक्लियोटाइड डिफरेंस. नाउ कम टू द टाइप ऑफ SNP. If you study the type of SNP, okay, so that is generally SNP occur in both the region, that is non-coding region and coding region of the gene. Now what do you mean by coding region? Means the gene that is code for mRNA and then further for protein. So that is the meaning of the coding region. So you can also observe the SNP both region, non-coding region and coding region. But non-coding region SNP that have no importance because there is silent mutation. Means we can use for the forensic purpose, but there no such type of effect can be observed in individual. So SNP, if there is more number of SNP in individual, it means the person is not healthy. There is no correlation between more number of SNP, person will be healthy. So just SNP give the clue or location or point or marker of particular gene, whether the defective gene is present in where. So that give the imprints, means address marker means what, that is the meaning. The marker means address, location, landmark, where the that with the help of that SNP difference we can locate the particular gene, the defective gene or the regulatory gene responsible for that defective gene in human beings. So try to know, gives the clue, the address, the tag. With the help of that we can find out the number of the gene, location of the gene, of the defective gene or the regulatory gene responsible for function of that defective gene that develop the disease in particular individual. किसी में disease develop हो रहा है तो ये किस लिए हो रहा है, क्यों? क्योंकि defective gene है, APO gene है, ठीक है ना। CFTR जीन है, उसमें डिफेक्टिव जीन है। सो दिस SNP मैप गिव द क्लू, गिव द एड्रेस कि यहाँ से इस पोजीशन पे आपका ये जीन है जो कि डिफेक्टिव। सो जस्ट गिव द location, address or landmark. Okay, so now come to the type. Just going to I'm just going to uh, that is the second type of SNP. One is that is the synonymous, and one is non-synonymous. What do you mean by synonymous? I'm talking about one is SNP present in non-coding region that have no importance, only for forensic purpose. Second, that is SNP present in coding region. In coding region, there are two types. What do you mean by synonymous, and what do you mean by non-synonymous? So try to know SNP in which both the form lead to same polypeptide sequence. अगर आपको पता होगा कि वन अमाइनो एसिड कोडेड बाय मोर देन वन कोडोन, ये आपको क्लास 11 में मैंने बताया है कि पार्टिकुलर अमाइनो एसिड सेरिन, एलानिन, एवेलिन कोडेड बाय 6 डिफरेंट कोडोन, 64 कोडोन आपको बताया है। अगर आउट ऑफ 64 3 नॉनसेंस है, ये तो बता चुका हूं। आउट ऑफ 64 61 codon that code for particular amino acid. Okay, if the same amino acid coded by different codon, means if nucleotide change occur but there is no change in the protein polypeptide amino acid sequence, then it is known as synonymous, means silent mutation, means no effect on the polypeptide chain. If there is no effect on the polypeptide chain formation, the same polypeptide chain is going to form, then it is known as synonymous. So such type of SNP is known as synonymous SNP, means change is there at nucleotide level but there is no change at polypeptide or amino acid level, that is known as synonymous. In case of non-synonymous, I'm just talking about the differ polypeptide sequence produced by non-synonymous, means missense and nonsense. So they difference in missense and nonsense. If a single nucleotide change lead to making different polypeptide sequence, from that point जहां से म्यूटेशन हो रहा है वहां से जो है कोडोन का, अमाइनो एसिड कोडोन का सीक्वेंस चेंज हो रहा है, देन दैट लीड टू मिससेंस म्यूटेशन। मैं चाह रहा था कि एलानिन वेलिन वेलिन बने, लेकिन बन क्या रहा है, एलानिन, ट्रिप्टोफान और टायरोसिन। So this is undesirable for polypeptide chain formation due to point mutation of one nucleotide और 2 न्यूक्लियोटाइड, नॉट 3 न्यूक्लियोटाइड। अगर 3 न्यूक्लियोटाइड का चेंज होगा तो आगे जो वाला जो पॉलीपेप्टाइड चेंज होगा वो सेम रह जाएगा, खाली वो न्यूक्लियोटाइड अमाइनो एसिड में चेंज होगा। सो आई एम जस्ट टॉकिंग दैट इज द जस्ट लाइक ए फ्रेम शिफ्ट कि एक म्यूटेशन हुआ और उससे आगे सारे के सारे जो अमाइनो एसिड गलत हो जाएंगे। जस्ट लाइक इन योर एग्जाम मल्टीपल चॉइस क्वेश्चन, आपने क्वेश्चन नंबर 5th का आंसर सी भरना था, आपने 6 में भर दिया। 5th का आंसर सी भरना था लेकिन आपने 6 में भर दिया और आप भरते जा रहे हैं। आप तो सही भर रहे हैं अपने मन से, लेकिन द आंसर इज नॉट करेक्ट। So this is type of missense mutation. And what do you mean nonsense? Means that is non-synonymous mutation always occur in coding region. Non-synonymous mutation always occur in coding region. So such type of change in which nonsense codon UAA, UAG, UGA will come. Then such type of termination of the polypeptide chain. Premature means जितना बनना चाहिए था उतना नहीं बन रहा है, only short nucleotide बन रहा है, तो उसको कहते हैं nonsense. Nonsense that is nonsense SNP, और एक है missense SNP, और एक पहले बताया मैंने synonymous। ये नॉन सिनोनिमस में 2 टाइप का बताया, मिससेंस, नॉनसेंस, और एक बताया आपको सिनोनिमस में दैट इज ड्यू टू साइलेंट म्यूटेशन। अब नेक्स्ट टाइप आई एम जस्ट कमिंग टू द फ्रेम दैट इज द पॉइंट म्यूटेशन। व्हाट इज पॉइंट म्यूटेशन? वन न्यूक्लियोटाइड चेंज। आपने देखा है सिकल सेल एनीमिया में पॉइंट म्यूटेशन होता है, ग्लूटामिक एसिड की जगह वैलिन हो जाता है, जीएजी चेंज हो जाता है किसमें? जीयूजी में। यहाँ पे क्या हो रहा है? जी ए क्या है? प्यूरिन। क्या है? प्यूरिन पैरामिडीन में चेंज हो रहा है। इसको क्या कहेंगे? ट्रांजिशन कहेंगे कि ट्रांसवर्सन कहेंगे? अगर प्यूरिन प्यूरिन में चेंज होता है देन इट इज नोन एज ट्रांजिशन, पैरामिडिन पैरामिडिन में चेंज होता है देन इट इज नोन एज ट्रांजिशन। लेकिन अगर प्यूरिन पैरामिडिन में चेंज होता है और पैरामिडिन प्यूरिन में चेंज होता है तो इसको कहेंगे ट्रांसवर्सन। So again we get the two different type of SNP. One is transition due to transition and second is transversion. So in case of transition, the same nucleotide, same means purine replaced by purine, one nucleotide in polypeptide sequence, in nucleotide sequence. अगर प्यूरिन प्यूरिन को रिप्लेस कर रहा है तो वन न्यूक्लियोटाइड को तो उसको कहेंगे ट्रांजिशन एसएनपी। और अगर प्यूरिन पैरामिडिन पैरामिडिन को कर रहा है तो उसको भी कहेंगे ट्रांजिशन एसएनपी। लेकिन अगर प्यूरिन पैरामिडिन के बाद से रिप्लेस हो रहा है और पैरामिडिन प्यूरिन से रिप्लेस हो रहा है तो उसको कहेंगे ट्रांसवर्सन एसएनपी, जो कि होता है किसमें? सिकल सेल एनीमिया। सिकल सेल एनीमिया में क्या होता है? ग्लूटामिक एसिड रिप्लेस बाय वैलिन। तो ग्लूटामिक एसिड का क्या होता है? जीएजी। सो व्हाट यू मीन बाय जीएजी? दैट इज द कोडन ए एंड जीएजी एंड देन वैलिन किसको कोड करता है? GUG. So A converted into U, only one nucleotide, and due to that change, there is change in the shape of RBC, the round shape to sickle cell. So this is also a, that is transversion point mutation SN. Now come to what are the different methods? Method of identification of detection of SNP. पहला क्या है कि आप डिटेक्ट कर लीजिए जो कि पहले से एसएनपी आपके इंडिविजुअल में है, किसी पार्टिकुलर ऑर्गेनिज्म में, उसको पर। और सेकेंड पॉइंट कि ऐसे एसएनपी जिसका पता नहीं चला है उसको भी डिटेक्ट किया जाता है थ्रू सीक्वेंस। ओके, सो थ्रू जीनोम सीक्वेंसिंग वी कैन डिटेक्ट द प्रेजेंस ऑफ एसएनपी or that is already known and the SNP that is not known till now. So there are two methods for identification of SNP with the help of sequencing. One is detection of SNP and other is identification of SNP which is not known till now. So SNP map just like a bar diagram in the genome that gives the clue for the doctor what will be the dose of particular drugs and what will the it will also predict the susceptibility of particular disease in particular person कि कितना ज्यादा सीवियर कंडीशन है और किस कंडीशन में किस तरह से ट्रीटमेंट किया जाएगा। so that things can be utilized that SNP map can be utilized for that purpose. okay, next डिटेक्शन कर सकते हैं नॉन जेल बेस्ड थ्रू जीनोटाइप टेक्नोलॉजी, हाइब्रिडाइजेशन विद फ्लोरोसेंट रेजोनेंस एंड हाई डेंसिटी चिप एरे टेक्नोलॉजी। सो दिस चिप मींस SNP that is the more advanced method for detection of particular nucleotide than the DNA microarray technology. So with the help of DNA microarray technology we can detect the limited number of gene, but in case of for SNP, but in with the help of SNP technique we can find unlimited, means more number of gene can be detected with the help of that SNP technique. So these are the application, and you can also study the SNP for the identification of the gene responsible for cancer. So increase the activation of chemical carcinogen that can be monitored with the help of SNP maps. So these are the vast application that is the SNP. So just try to know use and importance of SNP. What is the use? Means variation in the sequence. पहला मैंने आपको बताया वेरिएशन पता करना है किसमें? पॉपुलेशन का, इंडिविजुअल टू इंडिविजुअल क्या डिफरेंस है, वन न्यूक्लियोटाइड का डिफरेंस है। दूसरा कि आपको विद इन ए फैमिली आपको पता करना है कि क्या डिफरेंस है, और बिटवीन ट्विंस पता करना है क्या डिफरेंस है। सो वी कैन यूज द टेक्निक दैट इज SNP। एंड दूसरा how the effect of drugs on the different patients that can be easily monitored with the help of this SNP. And then disease development कि what is the condition of disease susceptibility कि ये susceptible है या नहीं है particular disease से, उसका severe condition क्या है, early stage है या later stage है या mid stage है, that can be studied with the help of this SNP maps. Okay, so we can also monitor the response to pathogen, means drug response to pathogen, the chemicals, drugs and vaccines on the pathogen. So SNP also throughout to key enable in the realizing the concept of personalized medicine, means person to person the dose of the particular drug will be vary, and that thing can be monitored only with the help of SNP. मतलब personalized medicine means the same medicine that is effective at different dose in different person, कि एक ही मेडिसिन है लेकिन अगर सपोज 10% है तो फर्स्ट पेशेंट में उसका डोज 20 mg होगा, दूसरे पेशेंट में 100 mg, तीसरे पेशेंट 150 mg होगा, उस तरह से इट विल वेरी अकॉर्डिंग टू सीवियरिटी. तो जो जिसमें कम डोज लगेगा दैट इज द मोर एफिशिएंट, मींस रिस्पांस इज बेटर. जिसमें ज्यादा डोज लग रहा है रिस्पांस इज नॉट बेटर. So such type of thing can be easily monitored, and it is easy for the doctor to prescribe the particular medicine of the particular dose with the help of SNP. Next, just to find out the susceptibility of the disease due to defect of particular gene or the regulatory gene, defect of particular gene as well as regulatory gene. Regulatory gene मतलब क्या है कि आप देखो आज कितने लोग आए हैं. अर्नब इज वेटिंग. कौन है अर्नब जी? हु इज अर्नब? अर्नब कौन है बेटा? ये बताओ अर्नब कौन है? हाँ, ये कौन है? So try to know what is the effect of this medicine, and what is the effect of defect of particular gene or the regulatory gene responsible for disease development. Such thing can be easily monitored with the help of SNP. Now write down the application. You can also write down the application of SNP. So SNP generally used for gene discovery and mapping. मतलब जीन का क्या फंक्शन है ये पता कर सकते हैं कि पार्टिकुलर जीन इज रिस्पॉन्सिबल फॉर व्हाट पर्पस, ये डिफेक्टिव है या नॉर्मल जीन है, और उसका सीक्वेंस पता करते हैं. दूसरा एसोसिएशन एनालाइज कैंडिडेट पॉलीमॉर्फिज्म, मतलब कि इंडिविजुअल टू इंडिविजुअल डिफरेंस पर्सन में क्या है एट द मॉलिक्यूलर लेवल. Such type of thing can be easily monitored with the help of SNP. And next is diagnostic and risk profiling. I am talking about diagnostic, means try to know the severity of particular disease. Ki with the help of SNP map, if the defective gene is there or the gene defective regulatory gene is there, such thing can be easily monitored with the help of that gene. With the help of that. So these things can be easily monitored with the help of SNP maps. So SNP map is just a vertical bar diagram on the line graph on the line, and that give the clue for the doctor for effective use of the particular dose of particular medicine, and to predict the severity of the particular disease. Next, response prediction. I have already explained this thing. Response of particular drugs, and then homogeneity testing, study designing. So बोलिए. Yes. नेक्स्ट दो कोई बाकी रह गया क्या? तो कितने लोग आए हो तुम लोग? बोलिए. हाँ, अब बताइए कौन कौन लोग आए हैं. गिव योर अटेंडेंस.
सर पंकज.
सर आई एम ऋतु गोलवाल.
क्या नाम है?
गुड मॉर्निंग सर, आई एम ऋतु गोलवाल.
लोअर क्लास नहीं है बेटा, ये
लोअर क्लास नहीं है बेटा, ये
तो पहला धीरज.
बारहवीं.
पहला धीरज.
दूसरा नंदनी, तीसरा हाँ ये मैं नेक्स्ट है कौन?
उत्कर्ष।
उत्कर्ष, बोलते जाओ बेटा।
प्रतिभा
निधि, नेक्स्ट
जरा
नेक्स्ट
पंकज
नेक्स्ट। आप लोग ने क्विज कंप्लीट नहीं किया।
मैंने किया है।
पंकज हो गया
यस सर।
और उत्कर्ष क्विज हो गया। निधि और कौन कौन आया है?
जी
हाँ
प्रतिभा प्रतिभा
प्रतिभा, और रजी, और आर से और एक और है ना, एक आर लेटर से एक और है ना, कौन है? और कौन कौन है?
क्या
हाँ सो
क्या
बताइए। नंदनी हो गया। So must give your attendance. Sir is observing your attendance. Okay, and sir is also observing our class. So don't use any other word so that it can cause problem for you. इसी तरह से आप कोई ऐसा वर्ड नहीं ग्रुप में आप डालेंगे जो कि प्रॉब्लमेटिक होगा, और अपना अटेंडेंस दीजिए। फिर आप जो है अपना जो क्विज है कंप्लीट करिए, और होमवर्क जो दिया गया है उसको कंप्लीट जरूर करिए। ओके, थैंक यू।